Hi, everyone. It's a great pleasure to deliver this lecture. I'm really delighted to see so many of you joined this afternoon. Microscopy is one of the most fascinating areas of science. It has a history of more than 600 years. Across this remarkable journey, it has made revolutionary contribution to virtually every discipline of science and technology. It has changed the way we observe, understand, and innovate. Therefore, this lecture will be more educative than research oriented. I will focus on some of the key historical development and breakthrough moments that shaped modern microscopy. Microscopy begins with a simple question: how do we see? Therefore, before we understand why microscopes are necessary or how they reveal the fascinating world hidden from the naked eye, we must appreciate the remarkable optical instrument we carry with us every day. That is the human eye. Although we often take vision for granted, the eye is an elegantly engineered system that forms images, detects lights, and communicates with the brain to create our perception of the world. Understanding how this process works forms the foundation for understanding the magnification, lenses, ultimately the microscope. If you look at the anatomy of eye, some of the important components are cornea, which is a transparent dome-shaped surface covering iris and pupil. It is both a protective shield and the eye's strongest focusing element, contributing nearly 75% of the eye's total focusing power. Then we have pupil. This is the black opening in the center of the eye's iris that regulates the amount of light entering the eye, and its size is controlled by two muscles in the iris. Then we have lens, which is a clear, flexible, biconvex structure located behind the iris that focuses light into the retina. By changing its shape, it allows the eye to focus on objects at different distances, and it is essential for producing clear vision. And the most important part is now retina. Retina is light sensitive tissue lining the back of the eye that converts images into electric signals, which are then sent to the brain via optic nerve to be interpreted as vision. Therefore, all these parts and components of the eye, they work synergistically to create the image of an object. If you look at the optics of eye, you will see that to be clearly see an object, it has to have certain size or dimension. It has to be placed at a particular distance and visualizing angle. That's because the lens is not so flexible, and we are only able to see in when certain criterias are met. We can see objects, we can see details of the object, we can also see the motion, and we are able to perceive different color, distinguish color. And we have to remember that a significant population may also have near or farsightedness that can be corrected using specific lenses. Now, despite this magnificent architecture that we all possess, we have certain limitations. For example, we are only able to see and perceive within the visible region of the electromagnetic spectrum, say within the wavelength of 400 nanometer to 700 to 800 nanometer. That is because the component that we have in the retina. It's also important, it has an evolutionary and adaptive reason that most of the food and fruits are within this range. We can see all those colors. But it has to do also with the photoreceptors. We are not able to perceive the UV ultraviolet light or the infrared light. Rather surprisingly, some insects and bees they are able to perceive the ultraviolet light. They have a specific photoreceptor in this region. And it also has significance in collecting the nectar, which has an impact in the pollination. Rather, even more surprisingly, certain snakes are having the power to detect higher radiation. They can detect their prey through the thermal radiation that is coming from their prey. So the vision has limitation, but certain animals can overcome this limitation. They have this capability. So we can only perceive with a certain wavelength of light. We also have to remember when we talk about the color, significant population also has red green color or blue yellow color deficiency. I would call this as a deficiency rather than blindness. Then why we need microscope? The first reason is the limitations of the size that we can see. We can resolve. When I say resolve or resolution, it means that our ability to distinguish the two objects or details within an object which are closely placed. And we can see, human eye can see without any external aid about 100 micrometer. That is roughly the size of the diameter of human hair. There are numerous hidden wonders in this world, such as cells, bacteria, intracellular structures, other microscopic structure which are not visible to human eye. And microscopes are important for numerous science and technology development. They combine multiple lenses so we can achieve higher magnification. We can design new material based on their micro and nanoscopic properties. Then what is a microscope? The microscope has an origin from Greek words. Micros means small, scopian means to look at. However, all microscopes have few things in common. They all utilizes some light source, either light or in more advanced electron microscope beam of electrons. And they have a series of lenses, either glass or electromagnetic lenses. Then they have a detector or camera. So all microscopes have this formula. Originally, the idea of microscope is to simply magnify an object that are not visible to naked eye. But today, microscopes can solve the complete structure of biological molecules or virus particles within a few days' time. Therefore, a microscope is a scientific instrument that produces a magnified image of the specimen. It has the ability to separate the details in the image, and it renders the details visible to the human eye or camera or any form of a detector. Then what is a simple microscope? A simple microscope is nothing but a magnifying glass, a convex lens that creates a virtual upright and magnified image when an object is placed closer than the focal length. So this is the simplest tool that demonstrates the fundamental principle of magnification. Now, instead of a simple lens, if you add another lens, it becomes a compound microscope. Now, if you invert this, it also becomes a telescope. We have to remember telescope has revolutionized much rapidly, progressed much rapidly than microscope. Microscopes took almost 6 centuries. And that's exactly why we will look into the historical development. The first part, the evolution of microscope. Microscope starts already in the 13th century. The spectacle makers in the Netherlands were using some highly pure glasses and lenses. In 1590, then after a couple of centuries, Hans and Janssen developed a single lens microscope, which has the lens which can magnify up to 9 times the original size of the object. The beginning of 17th century, Galileo became very much interested in telescope. He also used the microscope, and this microscope was used by his collaborator and friend Giovanni Faber. He was not a scientist, but he was interested to look at objects such as plants and parts and draw their structures. And he was the one who coined the term microscope for the first time. Then later on, it evolved. There was a compound microscope from Robert Hooke, and then there was a problem. We will discuss about this. Then Anton Leeuwenhoek looked at the bacteria using a highly precise single lens microscope. However, in the mid and late 19th century, Hans Abbe and Carl Zeiss became very much interested in developing microscope. They also proposed the theory of limit of resolution, which is commonly known as Abbe's diffraction limit. And today, all the diffraction limit can be broken and it can be overcome. We have super resolution microscope. We can look at nanoscopic level. Here I am only looking at the light microscope. Maybe we can look at some of the examples historically. Robert Hooke first developed the compound microscope. As you can see, he used a lamp and a bottle of water or that can shine the light on the lens. And then you can see the objects such as corks. He draw really beautiful images with a very high resolution already in the 17th century of certain bugs. But the challenge was good images were formed only near the center of the lens. As you move away from the center of the lens, the image quality becomes poorer. And this is due to spherical and chromatic aberration. So that is where the single lens was developed by Leeuwenhoek. He had extremely small lens of 1 millimeter in diameter and was able to magnify an object up to 400 times. Remarkable. But the challenge is that it is highly laborious and it's very challenging to take this lens all the time and draw the image. Robert Brown then improved the compound microscope and showed that you can image cells. He has shown the nucleus in onion cells. Then Ansabe came up with a fundamental physical boundary for resolution in 1873. He proposed that 2 points closer than roughly half the wavelength of light cannot be distinguished due to light's wave nature and diffraction. So he proposed the formula where he said the numeric aperture of the objective lens becomes also an important parameter along with the wavelength of light. Now, if you substitute in this the wavelength of light, if we take a visible wavelength of light in the visible region of the electromagnetic spectrum, say 500 nanometer, then we end up around 200 to 150 nanometer resolution. So, how can we see objects below this range? That's where the next evolution of microscope and the light sources have changed. Today, if you look at optical microscopes, they have few things in common. They have objective lens, they have eyepieces, they have specimen uh, chambers or holder, condenser lenses, and a light source. You can take both dark field and bright field images. And this optical microscope nowadays evolved. They can overcome Abbe's diffraction limit slightly using different techniques, such as in this case, a super resolution microscopy, where today we can see various parts of the cell. We can look at the cytoskeleton by clever combination of using labeling of the cells using specific dyes and image processing. But what about looking at objects at atomic resolution? How can we see that? How can we see atoms? This is what happened after Abbe's diffraction limit experiment. And today I represent evolution of microscopy as more like a tree. as shown until abyss diffraction limit, it was slowly developing. But what happened after in the 20th century is microscope has branched into different areas, particularly focusing on application on specific field. And nowadays they start to converge again. And the latest here we can see from 1930 onwards, electron microscopy taking off, cryo-electron microscope, scanning, tunneling microscope, and in the 2024th edition of science, there is something called microscope. We are talking about looking at reactions and atomic movements in real time, in attosecond time. So we will look how this achievement or this revolution happened. I discussed about abyss diffraction limit, but what happened in the 1920s is the wave particle duality has come up. Louis de Broglie stated that in his PhD thesis, all matters including electrons has wave-like properties, and he derived a relationship between the particle wavelength. In 1927, Clinton Davidson and Lester German demonstrated this by firing electrons at a crystal, observing diffraction pattern, a phenomena characteristics of waves, not particles. So this is a groundbreaking discovery where they have shown that electrons also behave as waves. What does it mean? If electrons are behaving like waves, we can use electrons instead of light. We can remember how the abyss diffraction limit affects smaller the wavelength, higher the energy, higher the resolution. But how to do this? So the idea was that if you make a cloud of electrons, unfortunately electron then scatter in the air. You need a high vacuum. So if you have a source of electrons and then you apply high electron volt or accelerated voltage, you can make that electrons behave in vacuum much like a light. Now, instead of glass lenses, which cannot be used because of the high power electron, high energy dose, electric and magnetic fields could be used to shape these paths followed by electrons. So, this is an analogy to glass lenses we use in optical microscope to bend and guide the light. So now we can look at how this revolution was done. Electrons have particular wavelength, and the wavelength of an electron beam depends upon the potential that we apply, through which it has been accelerated. So now substituting the applied accelerating voltage with this equation, we can find out what is the wavelength that we can generate using electrons. So what it suggests is that if we have higher the voltage, the wavelength is smaller. So we can go back to abyss diffraction limit, the same equation. And today, routinely, electron microscopes, we look at how it works soon, uses 40 to 300 kilo electron volt accelerating voltage. So if you substitute 100, 200, 300 kilo electron volt here, then we end up getting a wavelength of up to 1.96 picometer. Now we can go to atomic level resolution. So with this, the first electron microscope was built by Hans Ruska and Max Knorr in 1931. There is a demonstration. The first working electron microscope was developed in 1933, and after 6 years, Siemens produced the first commercial electron microscope. And the first in 1933, Ansluska used two magnetic lenses and able to see the resolution same as an optical microscope. Use of a third lens increase the resolution by twofold in already a year later. Then how this electron microscope work? What are the component? You all know light microscope. These are the few things that are important in electron microscope. First is electrons, filament, electron cloud, anode plate, magnetic lenses. So as you all know, the electrons are, if you take any source and apply a high accelerating voltage, you can eject this electron. An analogy here is an electric bulb. If you increase the voltage, it will become brighter. If you decrease, it will become dim. In electron microscope, the source of electron is a U-shaped uh, filament. It can be tungsten, lanthanum boride, and depending on the type of microscope and source. The video I'm showing here is more for scanning electron microscope, but the generation of electron and guiding this electron remains the same. So now if you apply high accelerating voltage, you eject the electron, you generate a cloud of electron. Now when the cloud of electron is there, it scatters in the air. So if you confine it in a vacuum, still you need something else so that you can generate a beam out of this. Since electrons are negatively charged, you can use an anode which is positively charged. So an anode plate which will pull the electron towards the anode plate, and then you can use it to guide. And now you have to focus this light and guide by bending. And this is done using a series of lenses. These are magnetic lenses. What are magnetic lenses? These are nothing but coils. They do the same function as simple glass lenses. Now, once you have the beam of electron guided through the lenses, then you have your sample. This is shown here for a scanning electron microscope. We will look at it more carefully soon. And then once this beam of electron interact with your sample, you have many phenomena because it is an high energy beam. Some electron may be ejected from your sample, and so X-rays can be ejected. So then you have a lot of detectors. When the electrons are generated, you can detect this. Through this, you will know where your sample is, where your sample is not. Something very important is that in electron microscopy or any microscopy, for us to visualize or see an image or details, it has to be presented in a certain contrast and brightness. Same thing is here, where there is a sample, you will see a certain brightness. If there is no sample, you will see a dark area. And now we will move directly into scanning electron microscope. And for this, I really use this tool very often when I teach, which is a tool developed by Microscopy Australia and Thermo Fisher um, Scientific, and this is called Microscope. And I here made a quick overview. If you go to this particular website, you can get a set of samples which are actual microscope images. What I'm showing here is all possible variety of samples. You can look at it. The whole idea here is that it's very easy to understand how it works. So I always like in arthropods, particularly butterfly wings, because they're very colorful, and we can look at the reason behind this colorful visualization. When you go to this, the right side, what you see here is a cross-sectional schematic view of scanning electron microscope. So it has all those components that I previously mentioned. Now it is filled with the air. Now our first task is to find a sample um, and load a sample. And before that, we can uh, focus on this schematics. There in this case, we have here electron source, then we have an anode plate, then we have lenses, and then we have deflector. then the sample there, and the detectors on the both edges. Our first role is to evacuate, and that means, and then now to use the way accelerating voltage. We can use around 10 kilovolt in this case, and then we can select a spot size. That is, you can change and check how the spot size affects. And then we can also look at the working distance, which is the distance between sample, and then we can switch on. We can closely watch what happens when we switch on. A beam of electron generated, guided to the sample, the deflector, now it is scanning. And in this case, you don't see much in the background. The idea is that, as I said, we have present the image in particular brightness, certain brightness and contrast. As we change the brightness and then maybe slightly the contrast, you start to see that a beautiful butterfly wing structures are visible. Yes. And then we may have to also slightly focus this and then change the brightness. Now you start to see the scales of a butterfly. We can also then change and play with the brightness and then change the magnification, look at the more beautiful structures. So this also help you to understand without a need to memorize any components or working principle of this. If you increase the magnification, you start to see more details and further details. Now you start to see this more stripe, and then you start to see even more the nanoscale structures. So the butterfly wing is an example for a structures which goes from macroscopy to micro to nano level. It's an hierarchically assembled or structured materials. And these are the structures of a butterfly wing that you see. In this case, what you see here is that where there is a bright, there is a very high material content. And for scanning electron microscope, you need a conductive material or a conductive coating, such as gold, iridium, or carbon, um, or any sources, a very thin layer of that, because you need to generate the secondary electron or backscattered electron. So now we can also define scanning electron microscope. What is scanning electron microscope is nothing but a simple analytical tool. It involves the generation and evaluation of secondary electron, to a lesser extent backscattered electrons. We use a finely focused beam of electron, typically 10 nanometer or less. As a result, we are able to get high resolution and high depth of field imaging. Now, if we compare light microscope and scanning electron microscope, as I said in the very beginning, all microscopes have a very similar way of arrangement, except the source of light, detector, and imaging principle or image formation principles are different. Scanning electron microscope is used to study almost any type of materials today, from imaging. Here I'm showing the capability of this to look at those hydrogels as an example. For naked eye, it looks highly transparent, but we see nearly 10 or less than 10 nanometer um, uh, fibers in agarose hydrogel. Here I've shown collagen fibers. Um, Here is a porous materials, which is aerogel, where you can see the hierarchically porous structures. If you take a simple organic molecule and it forms different type of gels in different solvent, it also has a different structure. You can clearly visualize this kind of nanoscale structures. In the left side, you can see screw-like, or here more like twisted helical structures. Um, Now I will move to the next topic, which is even more interesting, is transmission electron microscope. We have seen light microscope and SCM difference. Now we move to something even more challenging, As the name suggests, it involves transmission. I also want to stress there is also scanning transmission electron microscopy, and which is the same. It works somewhat similar principle. In this case, unlike the secondary electron, here what we see is the electron beam is passing through. As you can see, the instrument itself look very complex. A beam of electron hits your sample and then transmit. That means your sample has to be electron transparent, has to be very thin. And when a beam of electron interacts with the sample, there are many different phenomena are possible. The lights emitted, X-rays, other electrons, backscattered electrons. This is where we can do scanning transmission, electron microscopy called STEM. You have elastically scattered electron. It helps us to look at diffraction. Inelastically scattered electron, which helps us to energy expressive loss microscopy. And the actual TM image is transmitted electrons. We can see how it works looking at this very simple demonstration. Again, the same TM simulator. Here is the schematics of a transmission electron microscope in the cross-sectional view. In this simulator, it always has instruction appearing at the top. At the same time, you can also see that the button you have to press always highlighted with the yellow. So we can first go here and start by removing this holder. As I said, the transmission electron microscopy, and the beam of electron has to pass through this. So we can remove the holder, and then we can choose a sample. I like nanoparticles, but you can choose anything. This is a very good example to see. And now once you take the nanoparticle, these are loaded with the TEM grid, and it's now loaded to the holder. And now we insert the holder. Now once inserted, you can closely see what actually happens here. It evacuates, and then we can select certain accelerating voltage. In TM, it is anywhere between 80 kilo electron volt up to 300 kilo electron volt. We can choose 200 kilo electron volt. That is the option available. Now closely watch what happens on the right side when you switch on the beam. And behind this diagram, we also see the screen, fluorescent screen, where the beam of electron. Now one thing is that this beam is offset, so we have to bring it to the center. Now by bringing it to the center, using this beam shift, here you use, of course, this button, but in principle it works exactly same also in actual instrument. And what you see, these grids are the metallic grids, which can be copper or gold. And it also has a support film, can be carbon, some firmware, or it can be even graphene or silicon nitride, very thin, 5 to 30 nanometer thickness. There are more than hundreds of these kind of grid areas present in this. Now we can switch on the diagram because there are many aperture, and we can choose first the condenser aperture. You can closely see what happens when we insert the aperture here. Then we can choose the correct size of the aperture. This is very important. And when we choose, we can say use small or medium, and we can watch what actually happens when we choose this. And now we see when you choose a medium aperture, the beam is now not circular, and this is called stigmatism. We have to make it astigmatic by using these knobs here. And our duty is to first keep it to the center and then make it circular. We can go, and yeah, and we have to spread. And now once it is done, we can choose a magnification. The correct maybe start with low. Now it looks bit blurred. So that means maybe we also need focus. Before focusing, we also have to select and put the correct height. So Z height to make sure that sample is in the correct position and there is no more wobbling. This can be corrected as we are looking at the Z height. So we can correcting using this straight position plus or minus Z. And now um, you can make sure that everything is all right. And then maybe we look at the diffraction pattern of this. And now we are almost all set, but we have to still insert the aperture. So maybe the objective aperture now, which is somewhere here, and insert. Yes. Now we are almost set for imaging, but now we have to move this very close and to the center using this beam current. Yes. Now it is ready for imaging. So we can simply press imaging. So we will see our nanoparticles. Now you can choose the area of interest that you want if you want. So we can bring it to the center. Um, and then we can increase higher magnification, can be medium. And then if you want to look at this, we can go to the medium and then very high magnification where we can see um, high resolution image. Yes. So we can already see the individual lattices in this case. So now you also learn how transmission electron microscope works. The so it is if we want to define now transmission electron microscope, it's nothing but an analytical tool in which an electron transparent sample, so that is the key here, is bombarded with electron beam. So now what we really see is the intensity of transmitted electron, which is determined by scattering phenomena. In the interior of the sample is recorded. So that means because electron beam is going through the sample, so we're looking at the internal structure of the sample. That's why it has to be thin. It provides a very high resolution image of the micro or nanostructures, nowadays atomic. If you look at the complexity, how it changes, as we move on, the complexity and also the cost and the maintenance of this instrument increases. Uh, since I've been working on TM over the last 15 years, I will focus a bit more. Simply, if you want to make a TM sample, generally you have this kind of a grid which is 3 millimeter in diameter. Then we have extremely thin support film, can be carbon, it can be graphene, silicon nitride, 5 nanometer up to 30 nanometer in thickness. And some films also come with a variety of structure which are lazy, which has a random holes. You can have highly precisely defined holes on this. Also, that is meant for cryo-TM, we will see later. Very simple way of making is loading your sample. You need 1 to 2 microliter, and then you blot excess sample on the solvent. Then you have your sample ready after drying, and then it is goes for imaging. Now, TM has many advantages. We can look at structures at extremely high magnification, 4,000 times than optical microscope, 4 million times than naked eye. So we can get a finer detail. We look at internal structure, we can determine morphology. Today, we can look at elemental mapping, we can do in situ TM, we can heat the sample in situ and monitor. However, it has limitations, particularly when it comes to biological sample. The first thing is that sample has to be very thin and thickness 30 to 50 nanometer. Not the diameter, the thickness is important. 100 nanometers higher limit. That's because when you have internal structure overlaps each other, we cannot get good details. Sample should be mechanical robust because we are using extremely high vacuum. the tolerant to the electron beam radiation. Why? When your material interact with high energy electron, it can lead to electron beam damage, particularly um, when high dose is used. Um, One of the possible ways that an elastic collision, which can lead to diffusion, atom displacement, and vacancy creation in metals, inelastic interaction, radiolysis, which can lead to electronic excitation, radical formation, ion fragments, increase in temperature, and contamination. In biological sample, it's even more challenging because they are naturally in aqueous environment. The specimen must be dried before it can be introduced for imaging. It has to be organic, and unfortunately, there are no heavy element in biological samples such as virus particles, proteins. Ionization and burning is a common problem. However, and this limitation can be overcome by using negative staining. It's a process in which the specimen or the object that you're looking at appears as a light while the background appears dark. This procedure utilizes some heavy metal staining such as uranyl acetate or formate. Uh, this will give you rather good image. However, the challenge is that if you work for biological sample, you can get the structure, but you cannot distinguish some of the secondary structures of proteins. And secondly, anyway, it is a dried sample. This is not the native structure, so you cannot get more insights. This is where the second wave of revolution comes in electron microscope, looking at structures in 3 dimensions. So now I will move to the seeing is believing, the original title of this. Let me show you this image. And when I show this, very often people say the left side, it is a couple dancing, and the right side is Michael Jackson dancing. But if I look at this, in reality, these are nothing but some random stuffs placed. And depending on how you shine the light, you start to see this kind of object. Let me give another detail. In the left side, you see the image of an old man with a stick. In the right side, a small child with a spoon. But in reality, these two come from exactly same random object, depending on from which side the light is incident. If the light is coming from one side, then you see as an old man. So the problem with these images are they are all two-dimensional. How it is related to transmission electron microscopy? If I take a three object, a sphere, a disk, and because it's a transmission, and I get a conventional TM image, I will end up getting simply a 2D projections of 3D objects. Now, if I rotate them, sphere will look exactly same, but the disk and the line rod will look different. That means 2D projections depends on the orientation of the object. So I give a simple example here of a DNA origami structure is highly sophisticated design. If you look at the image in the right side, you will see mostly one type of structures, but you also notice some strange structures here. And they are not a different structure. It's not a different size of particles. It's exactly the same, but these two particles are placed down. So they are standing up, and that's why the image looks quite different. So, what it means, we cannot get all the details from simple 2D imaging. So, there are two ways to get three-dimensional structure. The first one is a single particle reconstruction. And this is where the revolution in resolution started. The cryogenic transmission electron microscopy, and the second one is electron tomography. To give the importance of this technique, I will start with a story. What happened in 2014 in the Natal region of Brazil. The inhabitants showed some unique symptoms. They experienced flat, pinkish rash, bloodshot eyes, fever, joint pain, headache. By May 2014, it was found that it's a Zika virus infection. It has spread to um, 50 countries, and children were born with microcephaly, that means a deformed head. Zika virus structure was solved in 2016, and a more high resolution structure was reported in 2017. This structure was solved not using any type of crystallography, but using electron microscopy imaging. And this high resolution structure was significant because this allowed to understand the structure binding sites. They helped us to tackle this and develop antiviral uh, drugs at the same time, also to develop vaccines. We can come back to the 2019-20 story. We all experienced the COVID-19, where in 2019 December, a strain pneumonia with unknown etiology was reported in Wuhan. In January 2020, um, it was confirmed that it was a coronavirus. In February 2020, a preprint showing 3D structure of SARS-CoV-2 spike protein, which is the same used for mRNA vaccine, was reported. And then March 2020, the paper was published in Science. Just within 2 to 3 months, the structure was solved, unlike Zika case, which is 2 to 3 years. That is the real revolution in microscopy. This has to do with the three gentlemen here, Joachim Frank, Richard Anderson, Jacques Zubosset. They have contributed in various aspects of three-dimensional structure solution. And what is cryo-TM? The very first cryo-TM was um, reported by Taylor and Glaser in 1974. They used the same specimen chamber, but using liquid nitrogen, and they used the most standard catalyst crystals frozen conventionally. Unfortunately, they got a lot of ice crystal, but still it was the first cryo-TM image, and what they have shown here is a diffraction pattern. So how cryo-TM is evolved, and what is cryo-TM? We'll come to that. So if you take a water molecule and freeze it normally, it forms hexagonal or cubic ice crystals. But if you take same water, freeze it rapidly below minus 144 degree, you get an amorphous solid phase, which is a glassy state that is not crystalline, but it's a transparent. However, to do this, liquid nitrogen is not suitable. Now, if you take a liquid ethane or liquid propane and rapid freeze, because liquid nitrogen is not a good conductor of heat or electricity, liquid ethane is a good conductor of heat, liquid propane is the same, you can use separately or mixture of them, then you can get a very thin ice. And this leads to what we call a vitrification. That means you can vitrify water by cooling so rapidly that it cools about 20,000 degrees Celsius per minute, then it forms a glass. This entire process of rapid cooling of any liquid medium in the absence of ice crystal is called vitrification. And how it is done, and when it is done, particularly when you have biological sample in water, you cannot take up on that. You can use water or buffer, and it is done in this way. And this is a specimen preparation setup. You have a bronze cup, and then you have a spindle and that helps to transfer the temperature from outside to this holder. Here is a liquid nitrogen poured so that it becomes completely cold below close to liquid nitrogen temperature. Then you pump liquid propane or ethane, you gaseous ethane or propane, you get liquid nitrogen here. And then use a device called Vitrobot because it's a robotic vitrification process. You take the same TM grid that I have shown in previously for blotting, and then you add the sample here about 2 to 3 microliter. Here are these filter papers, and this automatically blots, and that makes an extremely less than 100 nanometer thin ice where the sample is embedded in the ice. So Therefore, the eyes act as a matrix here. Now we can take this and you can transfer to the instrument and start imaging. So how exactly it works? Now here is a TEM grid, and if you take this grid and look at it, as I previously said, it has highly ordered holes, where in that hole, you can see these are called holy grid, and this is a carbon support film which has the holes, and these holes is where the vitrified ice is formed, and within each of them there are your biological particles, or it can be materials any type which are embedded. Now it is so thin that electron will pass through. Now this specimen is also maintained in the instrument at liquid nitrogen temperature, and nowadays even liquid helium temperature. And now if you take this for biological particle such as virus, they are all of the same size, they are atomically precise. So this idea developed by Joachim Frank is that if you take 200,000 to 1 million particle of same type, you can maybe um, get three-dimensional structure. Why? Because when you freeze, the particles are randomly oriented. So if there is a certain symmetry, these randomly oriented structures can be categorized into different groups. It gives all possibilities structures for this particular particle. This is a video from Gabriel Langer from Scripps Institute. He has shown exactly how Cryo-EM works. And here is a append off with their particles. This is a Groel chaperone complex. So you can imagine that in water they're moving randomly. Now, when you take this sample and you do this vitrification as shown before and go to imaging, if you they are rapidly frozen as shown below, different orientation. Now, I showed previously that 2D projection depends on the orientation particles. Now, if they're oriented like this, when the electron beams shines on this, they're all same particle. They're oriented differently. Now, you can get these different images. And now you can see here, there are like different particles coming from different orientation. You can identify similarly looking particles such as here, and this particle can be then collected together. This is called a classification. Once you classify into many groups, you can get an average of this. Finally, you can combine all this and get the final structure of this. This involves a lot of image processing, computation, and dedicated softwares and skills, and also use of protein database. So it's the whole sequence, much more than this. So I have shown previously the SCM image of these gels. Here is a Cryo-EM image of this. We can also follow the self-assembly and material transition with aging, or by hybrid materials, mixing biological particle with synthetic particle. This is a literature example to show that negative staining and the cryo-EM of T4 bacteriophage. Now you may ask, is cryo-EM is only for biological sample? No, you can use this specimen preparation technique, not necessarily for structure solution, but to understand the challenging samples such as battery materials, where some of these electrochemical processes are highly dynamic. It is still a challenge to understand the charge discharge cycles in real time. Conventional TM is unable to preserve the native state because these materials are highly sensitive. So there is a beautiful paper in 2017 that has shown how we can use sensitive lithium based battery materials. This shows at the top how the conventional TM, non-cryo TM samples, they burn within few seconds. Now, under cryo-EM sample preparation, you can see the atomic level resolution. Not only that, you can look at all possible grain boundaries defects within a material. You can also look at how the material changes with the time. So the impact of cryo-EM is massive. Now I will move to the final part of this is the tomography. I already showed you the images of the shadows. Now it's the same in TEM, but the solution for those is particularly useful for those particles and self-assembled structure which are not necessarily atomically precise but have very complex structures. And this is where the tomography comes. So what is tomography? Tomography is nothing but um, constructing higher dimensional structural details from a series of lower dimensional projections. In TM, we always have in conventional TM two-dimensional projections. Now, if we can use a series of these two-dimensional projections collected at different angles, tilted, then we can get a 3D reconstructed structure. That means we are able to reconstruct the three-dimensional structure of original 3D object. It has a series of steps. First, collecting a series of tilted images by tilting the sample, but it has limitation. We can tilt only to certain extent, and then construction. So how it works, because microscope is massive and highly sensitive, we cannot rotate the source and detector. We only rotate the sample. Here is a YouTube video that also shows how it is actually done schematically. So you tilt and you collect these 2D projections. Now, here is one of the examples of a gold nanoparticle self-assembly where we can see a selected images taken at different angles. And you can see the orientation. It is at almost 0 degree angle. It's a minus 69 plus 69. And here is the three-dimensional structure. And if I look at a number of images taken, we take about 1 or 2 degree increment. And this increment, the structures can be stitched together. It shows as if the structure is rolling in this particular case, but it is not. Once you have that, then you use computational reconstruction, and then you can color artificially here. Red means high contrast, which means maybe heavy elements such as gold. Then we go to green, yellow, green, blue, white, and then you can virtually take a cross section of this. Now, once you take this cross section, you can see what is the internal structure. We can locate every building block, and we can look at the coordinates of this. We can extract the three-dimensional coordinate and construct computationally the new structures. And we can distinguish internal morphology as I've shown you here, and two different structures. And we can also do what we can collect similarly looking particles and using sub-volume tomography, how an individual particle look like. However, electron tomography has limitations. The sample should be thin enough to obtain meaningful results. I showed the thickness and should be mechanically robust. But the thickness is fundamentally important here in this because as we tilt the sample, the thick samples will experience a increase in the size upon tilting because now the beam has to go through really a thick sample, even though it's 100 nanometer. When it is tilted around 80 degree, it will be almost 600 nanometer. It is really challenging to focus and find the correct balance and reconstruction. However, sometime tomography can meet all this, beat the formula. Here is a massive structure, more than 500 nanometer, and we can still get a very nice, beautiful structure and internal details. But there are other limitations, and because if you tilt on only one side, you also have missing wedge. There's one way to overcome this is called dual tilt, but the resolution will only improve by square root of 2. There are numerous other methods that are also coming, continuous rotation and getting better or devoid of any uh, missing wedges. Sometime you will see the edges are blunted, or that's because of the missing wedge, because we miss certain angles. I have recently written a perspective on this, which covers almost many complex structures and how we can observe what are the new details we can get in the morphology. I spoke about also the resolution, and here is a beautiful work by John Meo's group in UCLA, and he is working on atomic resolution tomography, where you can look at individual atoms. It's highly specialized, and instead of incremental angle that we use, he uses increment slope. And this is the same principle. You collect in different tilt angles, and then you construct 3D structure. This is very important for catalysis aspects. Other advanced methods include microelectron diffraction, where today we can see the atomic scale structure of protein less than 2 angstrom, almost coming close to the X-ray structure determination. With this, I would like to conclude. Um, I want to say that microscopy is a gateway to the invisible. Is more than just a tool for acknowledging images. It serves as a gateway to understand the hidden structures and processes that shape our natural world. Used in a diverse setting, from discoveries to teaching laboratory, research institution, and clinical environment, connects human perception to the invisible, micro, and nanoscopic world. It enables crucial discoveries and diagnoses that would otherwise be impossible. Thank you very much.